I like to think of the specific immune response as having three major steps. Step one is to create an army of cloned T cells. We need this army of cloned T cells to do step two, which is to activate our B cells and create an army of cloned B cells. We need our B cells for step three, which is making lots and lots and lots of antibodies that are going to clear the pathogen from our system. So those first two steps in the specific immune response require our T cells and B cells to bind to their one specific antigen. Let's look at these processes. So how do we create this army of T cells? We need the army of T cells to make the army of B cells to make the antibodies. So how do we get the army of T cells? T cells have to be activated through a process called antigen presentation. Once antigen presentation occurs, T cells will be cloned, and then we get our army of T cells. Antigen presentation is exactly what it sounds like. An antigen is presented to the T cell, and then the T cell has to decide whether or not that is the antigen that it was trained to recognize. Certain cells are going to act as antigen presenting cells. Typically, cells harboring pathogens within themselves will place antigens on their surface. So basically, if you have a cell that's infected with a virus, that cell will take some of the viral antigens and put it on its surface, presenting the antigen to the outside world. Another way we can present antigen is to use those phagocytic cells like neutrophils, macrophages, and dendritic cells. Those phagocytic cells gobble up the pathogen, digest it, and then take some of the little bits and pieces of the pathogen and put it on the surface of itself, presenting the antigen. So these macrophages and dendritic cells and pathogen-infected cells are floating around in our body fluids, holding out the antigens. And then basically thousands of T cells will come along reading the surface of those antigen-presenting cells. Eventually, a T cell comes along that can recognize that antigen. Once that occurs, we will activate our T cell. Antigen presentation requires the right surface proteins on both the presenter and the recipient. The cell presenting the antigen always will present antigen using something called major histocompatibility complex protein, better known as MHC. In the picture on the screen here, we can see our macrophage has those blue blocks on it, and those blue blocks are representing the MHC. The green thing is the antigen. So the presenter will always hold out the antigen with MHC. So let's say my hand is the MHC and my phone is the antigen, right? The recipient, on the other hand, is not going to use MHC to recognize the antigen. Instead, it will use a receptor specific to the antigen. In this case, the recipient is the T cell, and the T cell has on its surface that one specific receptor that has been produced to recognize this one specific antigen. Once the T cell receptor, there's my T cell receptor, that's my MHC, and that's my antigen. Once the T cell receptor binds to the presented antigen, basically, magic occurs. No, it's not really magic, but chemicals get released, the T cell is activated. Basically what happens then is the T cell divides, and then those two T cells divide, and then those four T cells divide, and we get this process of cloning. We're cloning this T cell because this is the one T cell that we know can recognize the pathogen that's in our body right now. So to kind of look at this process in a more pictorial format, right? Here's our T cell. Here's the fancy specific receptor on its surface. This is enlarged. This is smaller. It clones once. Now we have two, but then those two clone and we get four. Those four clone and we get eight. And you can see how we can very quickly create this very large army of T cells. Thousands and thousands of T cells, all with the same receptor that can recognize the antigen currently infecting the body. Let's take a look at this in a different way. On the screen here, we have our macrophage. The U-shaped things on the macrophage are non-specific receptors that are capable of recognizing a pathogen. We talked about these in a different video. They're called pathogen recognition receptors. The pathogen comes along and is recognized by the pathogen recognition receptor. This stimulates phagocytosis of the pathogen. The pathogen is digested. Now what we're going to do is we are going to use MHC protein to present the antigen on the surface of the macrophage. Basically, the macrophage is saying, hey, look what I found. Are there any T cells out here that recognize this? Because I found this, and it's holding it out on its surface. Now along comes a T cell. The T cell has a receptor on its surface that can recognize the antigen. If there's a match between the T cell receptor and the antigen, the T cell then will become activated, and that's when we get the cloning. Okay, we have an army of cloned T cells. But not all of the T cells are going to be doing the same job. Some of the T cells differentiate. So some of them become a little bit different and have a new job. We have cytotoxic T cells, memory T cells, and helper T cells. Cytotoxic T cells are a type of T cell that is capable of killing self cells infected with pathogens. So if we have a viral infection or if there is some sort of pathogen that's in the body that's growing inside of cells, the cytotoxic T cells are very useful because what they can do is they can recognize self cells that are infected with this one specific pathogen and initiate apoptosis in that cell. Apoptosis is programmed cell death. Basically, the cytotoxic T cell says, hey, you need to kill yourself because you're infected with a virus. Cytotoxic T cells can also release perforins like natural killer cells, and the perforins will kill the self cells. Other T cells become memory cells. Memory cells are really important if we become re-exposed to the pathogen. Upon re-exposure to the same antigen, the memory T cells are going to mount a very fast immune system response, and so fast that we don't even know that we were ever infected the second time. Helper T cells are also produced through differentiation, and the helper T cells are really important because they're needed to activate B cells.